so glad to have Scott today. He's been a friend to the foundation for a long time. Sarah will have his official bio and shares some of that with you, but just a quick story. I met Scott my first year. I've been here almost 6 years now, and he graciously toured his labs, and we figured out a new way to try to work together more intentionally. And he's a big part of the 3-burr strategy of breeding, biocontrol, and biotechnology united for restoration. So I always really appreciated his time to show me around. I had a lot to learn my first year, and Scott was a great teacher. So with that, I'll turn it over to Sarah. She has his official bio and all of his credentials, which are through the roof. And we're really, really lucky to have him today. So thank you, Scott.
Thanks, Lisa.
Take it away, Sarah.
All right. Scott Merkle is the professor and associate dean for research in the Warnell School of Forestry and Natural Resources at the University of Georgia. Got his BS in biology from the College of William and Mary and his master's and PhD in forestry at Virginia Tech, working in the area of forest genetics and tree improvement. He's been with UGA since 1987 and teaches dendrology and research in forestry and natural resources. What's not on here, Scott, is your prowess in dry comedy. And one of the main reasons I love listening to you, Scott, is because you're incredibly entertaining, and I'm really excited. You're like, oh God, but yeah, so I'm really excited to have you on here. I think you make cloning and embryogenesis and any topic that you have just fascinating. So thank you for joining us. As always, if you need anything, let me know. Otherwise, I'm going to mute my video and screen and turn it over to you.
Okay, one question before you go. Are you going to be monitoring the questions?
Indeed, yeah, you don't need to check that. I usually try to keep everything until the end. If something really pressing pops up, I'll interrupt you, but otherwise you just focus on your presentation and I'll monitor the chat and the Q&A.
That sounds great. Thank you.
Okay.
Yeah, and if they're not related to your topic, we can just type in an answer and we'll just keep it flowing. Sarah's good at facilitating, so take it away.
Sounds good. Thank you. Thank you, Lisa. Thank you, Sarah. So before I start sharing my screen so you can see my presentation, I'd like to welcome you to my luxurious Associate Dean office suite here at the University of Georgia. I've been Associate Dean for Research for 7 years now, and I figure that's enough. So I'm actually stepping down from that position at the end of this calendar year, the end of December, and I'll return to full-time research, a lot of it with Chestnut, and teaching. It's been a lot, no, I won't say it's been a lot of fun. It's been interesting and time to do something else. Okay, so I will start my screen share here, and hopefully I will pick the right thing to show you. Hopefully you see the presentation there. It looks like it's the right thing anyway, with the title and my name and a picture of a little chestnut tree in what looks like a peat plug. I was really happy to see this photograph used, I think, in the first of the 3-burr articles that came out in the Chestnut magazine, because this is something we produced in my lab, and this is a little chestnut, what we call a somatic seedling, and I'll define that for you soon. And this was a test we did. We actually published this little short note in the Chestnut magazine some years ago, where we figured out how we could germinate our chestnut somatic embryos in a preformed, it's not really a peat plug, it's actually a polyurethane and peat mixture. And we could actually autoclave this, germinate these little embryos under in vitro conditions, and then pull them out, rinse them off, and pot them up and get chestnut plants that way. So that's what that photo is showing. I didn't take this photo. I think this was a professional photographer came in and took this. Okay. So let me move this little bar out of my way here. That's better. So I'm going to kind of take a step back before we get into embryogenesis and cloning and all that stuff, and talk about, how to make chestnut trees. Not that I really am good at it, but pretty much I think most of you, if you know what the Chestnut Foundation has been doing the past 30 or so years, you know that the current technology that's being used for producing blight-resistant trees, or to get trees to test for their blight resistance, is the hybrid backcross breeding program, which produces at its end currently nuts from what's called half-sib technology. So those would be open pollinated nuts. The trees have been selected over the years during the backcross breeding program. We let them pollinate naturally and then collect the nuts and plant them out. That's called half-sib technology. Sib stands for sibling. So they're like brothers and sisters, all the trees that come out. You don't know when you plant a nut, you know who the mother tree is, but you don't know who the father tree is. It could be any tree that produced pollen that landed on that flower that made that burr and those nuts inside. And currently, that's how most of the trees produced in the Chestnut Foundation, that's where they come from. Now, tree programs that do a lot of tree breeding, especially like for commercial trees like loblolly pine here in the South and other trees, they have mostly gone to what's called full sib technology. And this is where you know both parents. So it's like real brothers and sisters here, not half brothers and sisters. And the way to do this is to take pollen from one tree and stick it directly on the female flower of another tree. And then you know both parents. But if you had to do this flower by flower, you'd never get done and you'd never make enough nuts. So they've looked into what's called controlled mass pollination, where they have equipment that just blasts the flowers with pollen. And they can make a whole lot of seeds that way, where you know that both parents are superior and the stuff you get is superior. Now, I don't know, of course, controlled pollinations have been done with chestnuts for a long time. That's where you use the pollination bags. But as far as scaling that up for mass production of nuts, that remains to be seen whether that will work with chestnut. Now, what we're getting into here, though, is a different technology. This is using clones, or the more friendly name is varietals, because everyone likes, varieties, right? But clones sound a little scary. I still use clones myself. And there's two main routes for doing that. One is called macropropagation. The most familiar type of macropropagation is rooted cuttings. And this is stuff you can do in the greenhouse or even in the field. You can make rooted cuttings, as I'll show you. And I'm going to talk about what's happened with chestnut making rooted cuttings. And in vitro propagation, in vitro literally means in glass. So this is doing stuff in sterile culture, in test tubes, Petri dishes, or I'll show you the vessels we use called magenta boxes. And this is called micropropagation in the broad sense. Okay, so there's macropropagation, micropropagation, and essentially there you are cloning. You're making more copies that are exact genetic copies of the starting material. Let's see, there we go. So this is way too much text for one slide, but too bad. I had to get it all on there. And I wanted to show you some pluses and minuses of going to clonal technology versus conventional seedlings. The advantages are on the top here. And there's more advantages in this, but these are the main ones I think we'd be interested in. First of all, if you can clone a superior or elite chestnut tree, you capture all what are called the components of genetic variance. And if you just want to think of those as all the superior genes that tree you're cloning has, you don't lose any when you clone it. Whereas if you cross it with another tree, you're not sure if you're going to capture all the superior genes or not, because first of all, there is meiotic recombination that happens when the egg or sperm are made. So you might lose some genes there. And then it gets combined with some other pollen parent that might not have the genes you want either, or it might have some genes you want, some you don't. If you clone the thing, you get what's called clonal repeatability. That is, when you plant out all those trees in a big row and they're all one clone, and then you test them for blight resistance, if every one of them shows high blight resistance, you can be very sure that genotype that you cloned really has the genes you want in it. So that's what I mean by it's a very powerful estimator of heritability of the traits you want. So it is a wonderful thing to do. And I know Kim Steiner was highly in favor of using clones for clonal testing because you get this clonal repeatability. Now, that's great for testing, but where you really can make use of the clones is by applying what's called a very high selection differential. And I threw this little plot in here at the last minute. I couldn't find anything that had to do with trees or plants, so I actually had to, it said animals here, and I put in my own labels. So if you look at a plot, let's say these are a whole bunch of different chestnut tree genotypes. Some will be not a low number here will be maybe highly, let's say, have low resistance. Most of them maybe will be somewhere in the middle. And then way out on this end, you'll have the very best stuff. Now, this distance between the mean of the resistance of the genotypes and a higher level is called the selection differential. And so if you select stuff that's way the heck out here, you're applying a very high selection differential to just get the stuff you want. If you can clone trees, you can make this selection differential higher than what's shown here. You can just take the very best of the best out here, propagate those up, get a lot of trees that are the very best. That's what cloning can help you do. What are the disadvantages? And we'll be talking about these a lot. One is, you can imagine, it's very relatively easy to just collect a whole bunch of nuts off the open pollinated trees or even control pollinated trees, stick them in the ground and get a whole bunch of plants, a whole bunch of chestnut trees. That's not so with cloning. Rooted cuttings or the in vitro culture especially, they require a lot of labor and operating expense. They also require specialized equipment and facilities, as I'll show you some of the things that we do with the in vitro culture. Cost money. Propagules, you can't just like pop them in the ground and get them to grow. In fact, you can't just, in many cases, stick them in the greenhouse and get them to grow. What you have to do is put them in planting containers. So if things don't grow well in pots, then you got to deal with that. But you can't directly sow these things in the ground like you can with nuts. And then finally, as you'll see, success in doing clonal reproduction depends on the species. Some species very easy to propagate, even by rooted cuttings, some almost impossible. The age of the starting material, young stuff is generally easy to propagate. The older it gets, the harder. And then there's other factors too. I'll mention a few of those later. I wanted to show you a really nice example of how rooted cuttings have done an incredible job for one species. And most people don't think of pines as being readily cloned, but there is one species, there's actually more than this, but this species, which you may have heard of, you may not have, called radiata pine. This is a clonal forest of radiata pine. If you've been to California, you've probably heard of Monterey pine. They're the same thing. The species name for Monterey pine, which grows on the California coast, is Pinus radiata. So in New Zealand and Australia, they call it radiata pine. These beautiful trees are probably they're less than 20 years old. And you look how uniform and beautiful and straight they are. This tree is not even native to New Zealand. It's native to California, but it grows like crazy in New Zealand. And they figured out that they could clone their very best stuff by rooted cuttings. How do they do it? They will plant seedlings in a big, well, if you want to call it an orchard, a big planting, let them grow to a certain size, cut them off, and radiata pine, unlike almost any other pine you're familiar with, actually sprouts from the stump like chestnut does. And you can see most of these are making a number of shoots. These are called stool beds because often they'll make 3 or 4 nice shoots, and it looks like you've taken a stool and turned it upside down, and those are the legs sticking up. So these things are then the source for cuttings. And here's some of the different size cuttings they use here, that they can actually root in these rooting beds and make thousands and thousands of trees this way. This is a very nice system. And there are other trees that are easy to propagate this way too. For example, cottonwoods and true poplars and willows are quite easy to propagate this way. They root very easily and no problem. You can clone them that way and make thousands of them. How about chestnut? Okay, if Steve Barilovitz is on, he's familiar with this because he's tried this himself. But chestnut, as we say in the South, ain't radiata pine. It's tough to do rooted cuttings of this thing. Not that there aren't some reports out there, but generally you can get some nice shoots coming up. These are stump sprouts. These actually aren't the B3F3 stump sprouts that we got from Meadowview one time. These are from stump sprouts from some of our trees, but you can see the stump here. Beautiful, vigorous stump sprouts. And you've seen this in the woods probably if you've looked at chestnut that's been killed back by blight. It sprouts from the stump very vigorously. Well, we got about 500 of cuttings about 10 years ago that were brought down. We had a guy help us who was an expert in rooted cuttings of oak trees. He worked for a company here in Athens called Select Trees, and but he had retired from there. And he helped us treat these cuttings with rooting compound, potted them all up, and here's what we got. That's a root right there. It's not a very vigorous root, but take a look at the rooting percentage we got here, 0.6%. That is not what you call a mass propagation system. It's and this was, this was our first stab at it, but we didn't keep it up. This is something we wanted to try. Now there are other reports out there with much higher rooting percentages, especially in Europe. Rooted cutting of European chestnut is quite well developed. There have been some reports in American chestnut of being able to do this at a pretty high frequency, but the problem came with overwintering when a lot of the rooted plants did not successfully overwinter, and then most of them were lost. And I've not seen any recent reports that have improved on this very much. Maybe some of you are aware of them, but I haven't seen them. Okay, now we can go on to micropropagation, or in vitro propagation. And true micropropagation, remember I told you in vitro propagation is micropropagation in the broad sense. But you can actually break that down into what we call narrow sense micropropagation, which is axillary shoot cultures, organogenesis, which is adventitious shoot cultures, and somatic embryogenesis. Now I'll show you some micropropagation in a minute, but essentially these are some micropropagated eastern cottonwoods. And if you look very closely in the gelled medium here, and by the way, this is a magenta box because the name of the company that makes them is Magenta. This is a sterile culture with shoots that we produce from cottonwood trees, mature cottonwood trees, in fact, and they're rooting in this medium. You don't even have to treat them with any kind of rooting compound or anything. They just root all by themselves. So essentially what you do is you take shoots, you cut them up into little pieces, and each one, make sure each piece has a little bud on it. You treat them with a compound called cytokinin, like benzylaminopurine, and they make new shoots from those buds, and then you can elongate those and root them and propagate it that way. Those axillary shoots come from the axillary buds on the shoots. It's almost like doing rooted cuttings, except you're doing it in sterile culture. Organogenesis is a bit different, and I'll have a little bit I can say about that later, but this is using essentially pieces of a plant that aren't supposed to make shoots and convincing them to make shoots. Like a piece of a leaf. You don't usually see shoots coming out of a leaf, but if you treat leaves of some species, including eastern cottonwood, this works with this, with BA, a cytokinin, it'll actually make shoots right on that leaf. You elongate them, root them, and you get shoots, new plants that way. And then I'll give you a definition of somatic embryogenesis in just a minute. So here is some micropropagation. Here's that same eastern cottonwood, which works great. But again, American chestnut is not eastern cottonwood. You can make axillary shoot cultures like these here. This is a piece of a shoot with a bud, and you can actually multiply the shoots, elongate the shoots, cut them off, and root them. But it's not real easy, especially the rooting step is challenging. Getting shoots isn't that hard. Getting really nice shoots that aren't dying at the tips with shoot tip necrosis is hard. And rooting is tough. Now, depending on what you start with, rooting can be not that bad. If you start with very young material, rooting in vitro is relatively easy, but not guaranteed. The older the chestnut material you start these cultures with, the more difficult it is to root. It's also highly genotype dependent. Some chestnut genotypes, pretty easy to make shoot cultures and root them. Other ones, not so much. And as I kind of indicated, when you get to mature trees, it is tough. Now, I don't want to tell you that this is as far as this has gotten. There are some recent reports that have come out, like Christy Lovat and Daniel Donnelly recently published a paper. They're up in Canada, where they improved the system. Allison Oakes in Bill Powell's lab and Chuck Maynard's lab published some papers where they greatly improved the axillary shoot multiplication and rooting. So things are moving along here, and this is important because I'll show you later, even with somatic embryogenesis, we have to often rely on going to something like this to get plantlets out. Okay. All right. All right, let's talk about somatic embryogenesis, since that's something I actually know about. So we've been doing somatic embryogenesis in my lab for longer than I've been a member of the Chestnut Foundation. I've been a member of the Chestnut Foundation for 30 years and a founding member of the Georgia chapter, to tell you the truth. I started this work in 1984, which is even longer than that. And of course, the first tree I picked made it seem like it was really easy. This is yellow poplar, Liriodendron tulipifera. We could start embryogenic cultures from seed embryos, get beautiful, beautiful somatic embryos. You're actually looking through a dissecting scope here at these beautiful somatic embryos of yellow poplar. You can germinate them on a plate. They all germinate. They make nice roots. And you can actually take little germinates, not much bigger than this. This is only a 60 millimeter Petri dish here, and stick them in pots and they'll grow. And I thought, oh boy, we should do this with chestnut. It'll be easy. No, it wasn't easy. It was like killer. Chestnut has been the most difficult tree I've worked with for somatic embryogenesis. I mean, we've done yellow poplar, sweet gum, magnolias, black locust, pecan. All of them were easier than chestnut. But I'll tell you how far we've gotten over the past, well, almost 30 years of working with chestnut here. Yeah, 30 years of chestnut. So like everything else, somatic embryogenesis has its pluses and minuses. As I hope to show you, it's a very powerful combination to make with full sib breeding, since we start with seeds. And so you can cross whatever parents you want. We'll take those seeds and start embryogenic cultures from them. It has, as I hope to show you, massive potential multiplying power. Start with one embryo, you can get hundreds of thousands. And there's a lot of potential for scale up and automation there. I'm not going to show you the kind of scale up that we've tested, but it kind of works with chestnut. Needs more research. Something I really like is if it works the way it's supposed to, your propagules or your somatic embryos have tap roots. That is, they have a shoot pole and a root pole. They look like embryos in seeds, as the definition says, that I hope you read since I skipped right over it. But they grow like true seedlings if it works the way it's supposed to. You can cryostore embryogenic cultures and hold them forever. Well, as long as you don't run out of liquid nitrogen, and I'll show you some pictures of that later. And this is the target material by which you do gene transfer. So it's pretty important to have a working embryogenic culture, as Bill Powell did, to transfer the OXO gene into that material. There are some disadvantages, though, as I'll show you in the depressing part of the presentation, which won't be that depressing really. There are some major bottlenecks, mainly near the end of the process, to getting those somatic seedlings out. The other bottleneck or hurdle is that so far, no one has been able to start embryogenic cultures from mature tree tissues of chestnut, any chestnut, European, Chinese, American. So we can't directly clone the very best trees. We have to start with seed explants, as I'll show you. So we started working, see, I told you 30 years, we started working with American chestnut, and the next year I joined the American Chestnut Foundation in 1989, and we got our first embryogenic cultures established. Because I didn't really know anyone in the Chestnut Foundation at that time, we were actually going up in the Georgia mountains, up to Brasstown Bald, and other locations where there were still surviving American chestnut trees that got big enough to flower and fruit, and collecting the starting material, which are the nuts. And that was a lot of fun, but it got tougher and tougher to find trees that we could get to that had fruit on them. Look how long it took us just to get our first germinants from here. That's pretty embarrassing, isn't it? Yeah, quite a long time. I told you this was a tough one to work on. Then once we got them to germinate, they kept dying off on us. So it was not until 2001 that we were able to get them through hardening off and the greenhouse and planted in a field test. And then finally, we were able to start scaling things up with some suspension-based culture system that I'll show you. So we started out working not with the hybrid backcross chestnuts, but with pure American chestnuts. And luckily for us, we after a while, we didn't have to go up into the Georgia mountains and stumble around with pruning poles to get the stuff we wanted. We were able to start collaborating with Fred Hebard, who had some what are called large surviving American trees that he could send us stuff from. Fred Hebard in the Chestnut Foundation. And then some people in the American Chestnut Cooperators Foundation, Gary Griffin and Lucille Griffin, who's under the Amherst tree here. They knew where there were these large surviving chestnuts, and they shared some of that material with us, the burrs they could get off those trees. And so we could work with large surviving trees. Eventually, we got some material from other chapters of the Chestnut Foundation. This is the Adair tree in Kentucky that I think Sarah originally arranged for us to get material from this tree to start cultures with. And we still have those cultures, Sarah. They're some of those beautiful cultures we have. And they're still going and still are able to make plants. And this was like probably, I don't want to talk about time, but it was probably like over 15 years ago we got this material. So how do we start the cultures? Here's an immature burr. It turns out that only immature seeds in these immature nuts will work to start an embryogenic culture. We take those white chestnuts, clean them off with Clorox, rinse them off with water so they're sterile on the outside so we can cut into them. And inside are little seeds developing at this point. This is not a mature nut. And this is only about 1.5 millimeters across. This is an immature seed that comes out of this nut. Put it on a tissue culture medium. I'll show you in a minute what the recipe is for that. And instead of just one embryo coming out inside, or instead of germinating and making a little seedling, it starts cloning itself. And it's the plant growth regulator called 2,4-D in the medium that makes the embryo clone itself. So these are the first somatic embryos coming out in October. If we keep transferring to fresh medium every month, 3 weeks is actually better. Here's the remains of that seed, and now there's probably the basis for about a couple hundred embryos. And finally, if it's still growing like this by January, we say we've captured it. It's going to keep proliferating. And you can see we started with one seed embryo, and now there's probably about maybe 500 of these embryos. And this is just one colony on a plate full of these cultures. I think you can start to see the multiplying power of this technology. Now, when we actually, I should say, well, what's in this medium? This is called woody plant medium. That's the basis for it, developed by Greg Lloyd and Brent McCown in 1981 is when they published this. And you can see it's got all the things that plants like to grow: nitrogen, phosphorus, potassium, calcium, magnesium, trace elements, copper, boron, molybdenum, iron, vitamins. And because our cultures are not photosynthesizing, they need carbohydrate. So we dump a big pile of sucrose in there, table sugar. They like to have an organic nitrogen compound source in there. Glutamine actually works the best. That's what we use now. That's an amino acid. And that's quite a lot of it, but the cultures like that. We used to use casein hydrolysate. Casein is milk protein. Casein hydrolysate is chopped up milk protein into amino acids, but just this amino acid works better than casein hydrolysate. And then here is the plant growth regulator. Most of you, if I haven't already told you this or you don't know, 2,4-D is an herbicide, right? It kills plants, but used at 2 parts per million or 4 parts per million, tiny concentrations, it acts as a plant growth regulator called an auxin, and that's what tells that original embryo to clone itself. And then if you want to make gelled medium, you add some agar or phytogel, or you'll see we use liquid medium. So when we're ready to scale things up, we actually dump some of that embryogenic material into liquid medium, shake it on a shaker. It's the same medium as what I just showed you, except no agar. And when it grows up enough, we dump it through some fine mesh screens to get rid of all the big clumps. And if you look up through the bottom of this flask using a microscope, you'll see all these little things that look like light bulbs. Those are somatic proembryos. And they're still growing in 2,4-D at this point, but when we dump them out, we take away the 2,4-D, and that releases these proembryos to go on and develop into mature embryos. These are not quite mature, but they're getting there. But you can see the little cotyledons and everything. You'll also see that they're collected on nylon mesh. If you look very closely here, this is tissue culture basal medium underneath some nylon mesh, and this helps them develop fully on this nylon mesh. We pick those off and move them to a plate of fresh medium, stick them in the refrigerator, and if we're lucky, they'll actually start germinating in the cold, just like chestnut nuts do when you stick them in the cold, just cold stratify them. Then we can take those out, put them in a magenta box with a germination medium that contains activated charcoal. That's why it's dark, and that protects the roots from getting hit by light and turning brown. They don't like getting hit by light. So activated charcoal protects them. We grow those up. Eventually we can harden them off put them in pots, harden them off in an acclimatization chamber, and get chestnut plantlets. Looks really easy, doesn't it? Yeah, there's about 10 years of work just getting that going there on one slide. So it's tough. But I do want you to know something about these plants. They look okay, right? They kind of look like chestnuts, but they're not growing very tall. And we're going to come back to that. They have very short internodes. Okay, just because we have to mention it, this is how we use these cultures for gene transfer. What the Powell lab does and what they did to get the OXO, original OXO trees, like the Darling 58, was something similar, but we do our cultures in liquid, and we use what's called liquid selection. You probably know that we have to use an antibiotic to pick material that we infect with agrobacterium, hope we get the gene that we want in. But we need the geneticin, which is an antibiotic, to get rid of all the cells that didn't get the genes we want and prevent escapes. And to really do a nice job, we do this in liquid. And you can see all these cells are turning brown. That's because the geneticin in the selection medium is killing them. But you see these bright yellow clumps in here. They got the geneticin gene, and then whatever other gene is on that same plasmid, and that allows them to keep growing in liquid selection and doesn't allow these other guys to keep growing. We originally did this with a Gus reporter gene, which turns things blue when you feed it a little bit of a certain chemical. And this is just showing that, you know, they are expressing the reporter gene, and then we can pick individual colonies. Once you stain with Gus, you've killed this stuff, but there's other ones that are available. Grow them up in their own flasks, check those. They make blue pro-embryos, they'll make blue embryos. And if we stain these little plantlets, they all turn blue as well. This was the groundwork, and Bill did similar work before he started using the OXO gene to get the gene transfer systems working. They're quite similar. They have the same end result, but this was, for his lab and for my lab, another 10-year odyssey, essentially, getting this to work. Now, if we're going to do gene transfer with OXO or any other gene, or if we get into gene editing, you have to have something where you can generate whole plants from single cells, because the single cells are the targets for the transformation. And then you got to be able to grow them all the way up into populations of plants. So this is very critical technology, and the embryogenic cultures are what made it possible. That was the only way to get the genes in. How about cryostorage? Well, we have been cryostoring every culture or copies of every culture we've ever started from American chestnut or hybrid backcross chestnuts or transgenic chestnuts for the last 20 years or so. And it's not that hard to do, certainly not compared to gene transfer. Essentially, we take little samples of those embryogenic cultures, put them in these cryo vials. Oops, I got to go back here, sorry. I clicked when I shouldn't have. These little cryo vials, we stick them temporarily in these containers. They have the very sophisticated name, Mr. Frosty container, very scientific name. And these actually get stuck in a -80 freezer. There's some isopropanol in the bottom of this thing that essentially allows these to very slowly drop in temperature from room temperature down to -80 at about 1 degree per minute. So that keeps horrible ice crystals from forming and damaging the cell membranes. And then there's some cryoprotectant, DMSO in there, and some other chemicals. Then we pop these out of here, stick them in these cryo boxes, lower them in the liquid nitrogen freezer. And essentially, once they're down in the liquid nitrogen, they can sit there for 48 hours or 48 years, because liquid nitrogen is -196. It stops all the metabolic activity. And then when we pull them out, we have to wash off all the DMSO and stuff and thaw them out. Of course, when they come out, they look like hell. Kind of like Ted Williams' head. Didn't Ted Williams like freeze his head when he died or something? Never mind, we don't have to discuss that. In any case, they look terrible, but even after a week, you start seeing regrowth coming out of here. 3 weeks, they're growing more, and we can reconstitute the embryogenic culture and make somatic embryos out of it. Now, I'll just mention here that we just recently did this, or just recently pulled out some cultures that must be at least 10 years old, maybe 12 years old, for some Georgia genotypes that we started embryogenic cultures from and froze at least 10 years ago. The Walker County tree and a tree on Brasstown Bald. And we're working with Marty Cipollini. We're going to grow these up, make trees from them, and then we'll have, well, the real idea is that we'll have our own trees to transform with genes if we decide to do that, or do gene editing on for the Georgia chapter. And of course, we're willing to do this for any chapter that wants to have, their own American chestnut trees put into embryogenic cultures to use for whatever you want. And I'll be glad to talk to anyone about that wants to get that done. We do have quite a collection already, though, from several states, from Pennsylvania, from Maryland, I think from Michigan, maybe, from Maine, and a few southern states as well. Okay, so what about hybrid backcross material? Well, you probably don't know this unless you've read one of these germplasm agreements. And this is an old germplasm agreement that this is based on. I don't know what the current germplasm agreement looks like, but the original germplasm agreements that were drawn up had a paragraph in here that said, no tissue culture of American chestnut foundation material will be permitted. I mean, let alone gene transfer. And I beat my head against the wall for years with previous administrators of the Chestnut Foundation saying, gee, we'd really like to see if our embryogenic culture system works with hybrid backcross material. And they didn't want to deal with me. But first, when Brian Burns took over as president, and enhanced greatly by Lisa, we have been working with TACF material now for several years. And if you read this, this was a special clause that was put in that especially allowed our lab to start embryogenic cultures and work with them for research. And it's made just an incredible difference because we found out a lot about culturing BC3F3 and other TACF material. And it's just so great having a partner now to do this with the Chestnut Foundation. Okay, so did it work with TACF material? Yes, it does work. It doesn't work exactly the same. It turns out the Chinese chestnut component of the hybrid backcross material, even when it's 15/16 American, has an impact. There's some genes in there from the Chinese chestnut that don't act exactly the same way. You can see that the starting culture looks a bit different. It tends to make embryos instead of those yellow masses that we started with. And it's a little bit more difficult to maintain the cultures and to get plants out of them. But you can see with open pollinated material, we got 19 out of 20 open pollinated cultures started. And even with control pollinated material, we have been able to start cultures. This was our best year, 8 out of 9. Some years we have not had luck with BC3F3 material, but usually we at least get some going, and it makes beautiful plants, and it works a lot like the pure American chestnut. But I know now that the foundation is reassessing the breeding program, and it looks like we're going to be maybe making use of more material that is not 15/16 American. Maybe we might use some B2 material, or maybe even some B1 material if it has the characteristics that Jared thinks we want for the program. Well, this was some work done by Christine Holtz, who was a master's student in my lab, where we got pure American chestnut. This is all one season that we got this material. Some B3F3 material, some B2 material, backcross 1, some F1, and some pure Chinese chestnut. And you can kind of see as the amount of Chinese chestnut genome increases, we lose our ability to apply this protocol to start embryogenic cultures. Now, some of these are very small samples, though. We only had a few B2s and B1s and F1s and Chinese chestnut genotypes to try. We had lots of American chestnut and B3F3s. So it's not great, but it does give you an indication that some tweaking needs to be done if we're going to use material that's got more Chinese chestnut in it. Probably it's because Chinese chestnut and things closely related to it have higher endogenous auxin levels in them. And so when we apply a big blast of 2,4-D, they don't like it. They've already got auxin, they don't want any more. So let me show you one other thing. It turns out that by tweaking the protocol, we were able to actually start some cultures from Chinese chestnut, but not using 2,4-D. We actually had to use a weaker auxin called naphthalene acetic acid, NAA, and that gave us a few Chinese chestnut cultures. Unfortunately, this was the high watermark of our Chinese chestnut work. We were not able to get plants out of these, even though they looked pretty darn good. They would not germinate And we have not recovered plants from these cultures, even though Ryan McNeil recently busted his butt on this. Ryan's a member of the Georgia chapter and a board member who may be watching this for all I know. He works out at the Hort farm during the day though, so he might not be watching. But it means that if we're going to go to some of this other material that has more Chinese chestnut in it, we're going to have to tweak things and do some more experiments to try and get systems going. But overall, this is how I envision this working with the breeding program. Someone does the breeding, they send us the nuts, we start the cultures. And this was what we have done this with the B3F3 seeds. We get beautiful cultures going. We split those, make plants from some of it, cryostore copies of the rest of it, put the things out in the field or wherever, however you want to screen them, maybe with small stem assays or Phytophthora screening. And the ones that are the very best, we can go back into cryostorage, pull those out, clone them up. Ideally, that's how I wanted it to work. But you'll see there are these bottlenecks to deal with. And here they are. So everyone get ready for the depressing part of the presentation. It's not that depressing, but it is challenging. Let's put it, they're challenges, okay? They're not bottlenecks or hurdles. They are opportunities and challenges. And I'll show you a little bit about each of these and tell you what we might do about them. First of all, let's take a look at let's just list them first. It takes a long time, as you just saw, from culture initiation to somatic seedling production. I'll show you a full diagram of that so you see exactly how long it takes. We have a lot of embryos that you'll see, they don't look that great. And they don't germinate that great either. In fact, the majority of them don't look that great or germinate that great. I showed you just the most beautiful pictures though, because I'm the speaker. I can show you whatever I want, right? Okay. Somatic seedlings, we used to undergo, and I know Bill's lab did too, incredible losses when you're trying to harden them off to get them out to the greenhouse or to the field. I know Bill's lab has become a lot better at this, and so have we, but we've got a ways to go. And then once you actually get them in the greenhouse, they don't grow that great. They have a weak root system. They have short internodes like I just showed you. And we want them to grow more like real seedlings, right? And then finally, this inability to directly clone proven trees. So how long does this take? This slide, the last time I showed it, it said 2.5 years. But when we figured out just how long this step down here really takes, I had to expand it by another month and a half or two or so. So take a look at this. This shows all the different steps that I already showed you. By the time you have little plants, maybe foot tall, 2 feet tall plants to put in the field, over 2.5 years, much longer than conventional seedlings, which you can get like in a year, right? You can be planting this a year after you did your control pollinations, I believe. What a difference. But I will tell you this, once you have gone through these steps here, these cultures will last for years. And so you don't have to reinvent the wheel. Once you have the culture invented, it's just making the embryos and plants that remain. So it's not quite as bad as it looks. And you can make thousands of these. What about the embryo quality? I don't think I've ever shown you this slide before because it's so weird. These are our attempt at getting mature somatic embryos that resemble the zygotic embryos you find in a mature nut. The only one that kind of looks like it should is this one right in the center that looks like a turkey. So you can see it's fair size. It's not as generally as big as a full-size embryo in a nut, but it's pretty good. It's got the big cotyledons. It's even got a nice little root meristem here, and hopefully there's a shoot meristem buried down in here. But here's some that didn't do so hot. It looks like it's got a root meristem, kind of blobby. And then some of them just look like monsters. And maybe they have a meristem that you can find, maybe they don't. This one does right here. Well, when you do histology on these and look at what's going on inside, you see what the problem is. Here's one. Here's a cotyledon that's been cut through. This is a longia section. It's got a nice little closed vascular system developing, but not a very well organized root meristem. And maybe this is the shoot meristem here, but it doesn't look good at all. Here's another one. You can't even find a root meristem. Those are the cotyledons right there. No root meristem, and it's trying to make a shoot meristem. Most of the embryos that we cut didn't have a beautiful root or shoot meristem. In fact, the person who was working on this gave up before they could find somatic embryos that had really nice shoot and root meristems. But obviously they have these meristems, some of them, because otherwise they wouldn't make somatic seedlings. So what have we been doing? To overcome this, if we can actually get, in many cases, embryos that will produce a shoot, but no root. So we can harvest the shoots and micropropagate them. We can either harvest them and root them directly, or we can cut them up and make even more plantlets using micropropagation. And this is, I think, a standard procedure. I don't know if Bill and Chuck were still using this or not, but they actually deliberately don't try to germinate their embryos anymore because so many of them don't germinate with that taproot. They treat their embryos with cytokinin, benzylaminopurine, and get them to make adventitious buds on them, elongate those shoots, harvest them, and root or multiply them. Both of these things work for cloning up if your somatic embryos will not make a nice somatic seedling all by themselves. And so we do this quite a bit. I used to be such a purist that I didn't want to do this, but if it's the only way to get plants, we're going to do it. Here's another problem. The acclimatization losses we used to have. The solution we found was more patience than I have, but we have to do it. It takes a lot of patience while they're going through this step of hardening off. We have these dome trays where we control the humidity. This is perlite sand in the bottom of the trays. We dump a bunch of water in there. We keep them in the trays, gradually opening up these vents in the top. And what you don't see is this all these trays are inside another plexiglass chamber that keeps the humidity from dropping. We found out that if we give them 3 months, which is like 3 times as long as we give most of our trees that we produce this way, that is the other species, that we can get quite high survival, but we have to be really, really patient. And that's what we found out. And here's these trays with the domes off and then out to the greenhouse where they actually will grow quite well after we repot them. But it took us a long time to figure out why we were killing these. We were too impatient. Well, out in the field, you've probably seen this. Well, maybe you haven't. If you've seen some somatic seedlings planted out, this is the way they grow. They want to grow sideways, or they want to start with, or almost put on no length at all. Now, this one's been growing for a while. This is up at SUNY ESF. And what they've taken on as standard practice is they go ahead and cut this horrible thing off and let it resprout from the stump. And look at these shoots coming up. And then they select the very best one, cut away the other ones, and they can get a 6-foot shoot out of one of these, nice and straight with nice long internodes in 1 season. That's Andy there. In his younger days, he still looks like that pretty much. Well, here's some more things we're doing here. We found out that we can actually do this when they're still in pots. Remember these really short internodes? When they're a little bit thicker than this, that is, we can go ahead and cut those off. And that's what we've done here. It looks terrible, but we've actually cut everything off of these pots, let them resprout, and those new sprouts that will come up in the pots will grow just like seedling sprouts with nice long internodes. If you look closely here, you'll see where the sideways stem was, and we cut that off here and here. So we can overcome a lot of this problem we were having by managing them in the greenhouse this way. Also, I don't know if Ryan's on here yet, but we're also looking at enhancing the root system first before trying to get shoots to elongate on here. There's been some papers that have come out recently that if you manipulate the nitrogen in your tissue culture medium, you can get a nice enhanced root system. And then once you get it potted up, the shoot will take care of itself once you have the good root system. So Ryan's working on this right now. It's a crazy idea for us, but it seems to have worked with maybe some conifers out there. And so we're trying that. Finally, so I'm almost done here. I've probably gone way over my time, but Sarah hasn't cut me off. How about cloning mature trees? Well, I'm here to tell you it can be done, and it can be done using embryogenesis, and it can even be done in the same family as chestnut in the Fagaceae. There are at least 2 species of European oak now. Cork oak, Quercus suber in Spain and Portugal has been cloned this way, where they actually can take branches off of mature cork oak trees, put them in the greenhouse to get some new epicormic shoots on them coming out of those, coming out of their stems they harvest. They take the leaves off those shoots and they can get somatic embryos from them, which is amazing. In fact, I think there's even a 100-year-old cork oak tree that this has been done with. It's also been done with Quercus robur, the English oak or pedunculate oak, and maybe one other species. Not any North American oak that I know of yet. But there is hope to be able to do this with chestnut. We've tried it a few times, maybe kind of half-assed, as we say in the South, and we haven't gotten it to work yet. And we have not had time to go back to it, but it's a possibility. Something we have worked on in the lab, and it works great, is there are some species where you can take floral tissues or inflorescence tissues and start embryogenic cultures from those. Obviously, these are going to be from mature trees, since it's mature trees that flower. If you're not familiar with sweet gum, it's all over the place in the South. It's not like we need to clone them, but we have. This is what the male flowers of sweet gum look like. They look like a stalk of broccoli when they come out in the spring. If you go into the dormant buds in February, you can find these flowers. Of course, they're not expanded yet. And you can take a piece of that inflorescence, put it on a medium, and get somatic embryos to come out of it and proliferate like this. So that's worth trying with chestnut as well. And again, we have tried it in a little bit, hasn't worked yet, but you know, research is research. We can go back to it. Finally, we're just starting some discussions with scientists in the Chestnut Foundation and others about perhaps applying some interesting genes that have been known about for several years now, that if you can overexpress them in plant tissues, they will change the development of those tissues. One of them's got the name Wuschel. That's probably the most famous one. And I probably misspelled that. I think it might have a C in it. And these are meristem promoting genes that perhaps we could take callus started from a mature tree, which is not hard to get plain old, you know, callus that doesn't know how to make buds or embryos, and transform it with Wuschel or other genes and get it to make somatic embryos. It's a long shot with chestnut, but people have shown that they can get embryogenic responses in other plants by overexpressing these genes in the tissues of those plants. Well, so is that enough information? I hope so. It was so much that I originally had a summary slide, and then I thought, you know what? I don't want to summarize this. Let's just go to thanking the people that do the work, and then you can ask questions, and we can discuss things. Ryan Tull's been in the lab for about 10 years now, and he is the guy who runs all our culture initiations and all the plantlet production, and he's recently become a guru at transformation as well, although not with chestnut. We're doing some ash transformation. Heather Gladfelter, about this time last year, became Dr. Heather Gladfelter. She was a research professional in a lab, and now she's a postdoc. She did a lot of the transformation work. She's also an expert in somatic embryo technology and works with the embryogenic cultures and the shoot cultures as well that we have. Paul Montello is the guy who does all our cryopreservation, and he's been doing that. He's been working with me for over 20 years now. And Ryan McNeil is working on a PhD in the lab. He is doing different things with chestnut to try and improve the embryogenesis system for us, and he's been great. He's got a full-time job as the director of the horticulture farm, and as I said, he's a TACF board member. I want to make sure I thank the organizations and people that have helped us over the years. Of course, the Chestnut Foundation, the Georgia chapter. Brad Standback has been very supportive of our work. The Forest Health Initiative, which is actually over now as far as chestnut is concerned for the most part, but they gave us a lot of support. Arborgen originally supported our work going way back. And of course, the cooperators gave us material to work with from their trees, which they're very, very touchy about, but they still shared their material with us. And then people in the Chestnut Foundation, Jared, Fred, Sarah, Marty in our own Georgia chapter, the Griffins in the Chestnut Foundation, American Chestnut Cooperators Foundation, and then our collaborators at SUNY ESF that have worked with us over the years and we're still working with. So that's the end of my presentation, and I can, for now, I guess I'll stop sharing the screen unless we need to go back to any of these slides, but I think for now I'll just stop sharing it. So, wow, that was kind of long, wasn't it?
That was long, but it was great. Lots of really fantastic information, and I've got lots of questions here for you.
Great.
Let's see, where do we start? So Anita Klein asked a couple questions, and I think they might be related, so I'm going to throw them both at you at the same time. One, how susceptible is chestnut propagation to somoclonal variation? And if you treat chemicals that disrupt DNA methylation, can you get older chestnuts to make roots?
I don't have good answers to either of those. I have not seen evidence of somoclonal variation as far as the material we get, but pretty much I would imagine if there are somoclonal variants, they might be mainly things that, you know, don't make it through all the different bottlenecks of getting germinable embryos and growing plants and things like that. As far as the trees that come out the other end, we haven't really seen evidence of somoclonal variation, but, you know, there could be some. As far as the DNA, you know, demethylation, I have no information on that. I'm sorry. I know what you're talking about, and it's possible we could do that. We have some other things we're going to try as far as rooting and overcoming some of what we think are the barriers to rooting. I'm not ready to talk about those yet, though.
Some questions for clarification. Jim asks, plant epigenetic issues? Question mark.
Again, we don't see evidence of epigenetic you know, changes, but of course, you know, our trees mostly we've only got a few that have, you know, grown up to the point where we can actually see what their mature phenotypes look like. Bill has older trees that he's had in the field a longer time, and probably Bill Powell would be a better person to ask about those kinds of changes.
Meg asks, do any of the agrobacterium tools incorporate fluorescent proteins as markers?
The ones we have not used GFP markers. We've always stuck with GUS, and that's my personal bias. Originally, when Bill was developing their system, they relied on GFP as the reporter gene, and it worked fine for them. I've just always preferred GUS. I think it's because I have trouble seeing GFP. Sometimes I'm going, is that really glowing or not? Whereas, you know, the GUS, it's blue or it's not. It's my preference.
Okay. And then Scott asks, what is the OXO gene? And both Lisa and I put a little information in the chat, but I wonder if you don't mind, you know, real quick addressing that.
No, I don't mind at all. First, I have to, you know, give credit, all the credit to Bill for identifying this as a potential blight resistance gene, which he identified early on as a perfect candidate. You may know, or you may not know, that Cryphonectria parasitica kills the tissue. When it makes a canker, it kills chestnut tissue and eats the dead tissue by using oxalic acid. It puts out oxalic acid, it kills the living tissue under the bark, and then it eats the dead tissue. And that's what eventually girdles the tree and kills it. OXO stands for oxalate oxidase. It's a gene that is in lots of plants. The one that Bill's using is from wheat. It is a gene that actually breaks down oxalic acid. So Bill's idea was that if you can have the chestnut tree make oxalate oxidase, then when it encounters the fungus, and the fungus is making oxalic acid, it can actually break down the oxalic acid before it has a chance to damage the tree or kill it. And it does appear, when you look at the, you know, the cankers that are on his trees, it appears to be doing that job, because the cankers are in many cases even smaller than cankers on Chinese chestnut and heal over pretty rapidly.
Meg asks, have comparative transcriptomes or microRNA evaluations of chestnut?
Have compared? Well, there has been work going on with genomics of Chinese chestnut and American chestnut, and there have been comparisons of them, but I've not been involved in that. But I will tell you this: what they looked at that actually gave us about 30 different genes to transfer into American chestnut to test them for their impact on blight resistance is they looked at transcriptomes of cankers on infected Chinese trees and infected American trees, and then focused on genes that had different levels of expression in the Chinese cankers than in the American cankers. And we've been testing a number of those genes. Bill's lab has too. A few of them show some promise, but that's how we made use of the relative expression levels of genes in those two transcriptomes.
And that's being done again now too. Just, I know that's ongoing with some clones this year and next year, just doing some additional time course studies and some metabolomic studies as well. So, you know, that work is ongoing.
Mm-hmm.
Scott says, so basically with cloning, it means that one seed produces multiple seeds.
Yeah, I'm glad you asked me that question. So what we're doing right now, that somatic embryogenesis that I've described that we use is actually embryo cloning. We start with a zygotic embryo and get it to clone itself. But those last photos I showed you with the sweet gum and the oak, that is somatic embryogenesis as well. And that's real somatic embryogenesis, where you're taking cells of leaves or flowers and telling them, you're not going to be a leaf cell or a flower cell anymore. You're going to make embryos. And that's a very, very powerful thing if you can do that, because then you can clone mature trees.
That would be awesome.
Wouldn't it?
Robbie Shaw, what is the intent of the cryotherapy on breeding samples?
Okay, so if you mean the cryo storage or cryopreservation, there's a number of reasons for doing it. One is we wanted to get as many surviving American chestnut trees represented in our cryo library as possible, because as time goes on, we're losing, especially in the South, we're losing those surviving resprouting chestnut trees. So if we can conserve their germplasm essentially forever and bring them back out and grow them up, if we come up with different ways of stopping chestnut blight, and we will still have the germplasm from those trees. Number two is we have thousands of embryogenic chestnut cultures. Well, not thousands. We have thousands of embryogenic cultures. I would say we probably have over 1,000 embryogenic chestnut cultures. If we tried to maintain all those cultures continuously, we'd never do anything else. In fact, we'd never be able to maintain all those cultures because they have to be transferred to fresh medium every 3 weeks. We need an army of technicians. Plus, they lose their ability to make somatic embryos over time, over 10, 15 years. They won't make embryos anymore. So we put them in cryo storage, and pretty much the way they go in is the way they come out. It's like they haven't aged at all if we can cryo store them.
Okay, let's see. Is the Wuschel gene complex like stem cells found in animals?
I can't answer that question. I'm sorry. I've read a few papers about Wuschel and some other of these meristem genes, but I do not have expertise in where they stand as far as comparing to other genes.
Yeah, I can't help that either. I know Debbie's on here. I don't know if she.
Oh yeah, maybe Debbie can answer that.
She can answer that, but let's see. Jim C., do you have papers on your cryopreservation? I want to learn more.
Yes, if you want to email me at smerkle@uga.edu, SMERKLE@uga.edu, I can send you PDFs of our cryo papers.
And let's see here. I think this is John Wenzel. He said, Scott, because you can get so many starting points from the culture, why shouldn't we create our own labs to grow out the embryos you could provide, and we do the work to get them up to actual saplings?
I would be glad to do that. I will send anyone who wants an embryogenic culture, or even one where we've produced somatic embryos, so it's already making embryos. It's not just the proliferating culture. To anyone that wants to carry them the rest of the way, even if it worked a lot better, we only have so much room to germinate embryos and to grow them up into plants. We'd be glad to send stuff out to people who want to work with the cultures, or we can bring them to whatever point they'd be useful for you and then send them to you.
Let's see, and then one last question then. I hope I don't butcher his name, but Burak asks, did you try to juvenile mature plants by any methods?
Oh, to rejuvenate mature plants. Yeah, so pretty much that's what we were doing. When you, for example, cut off a tree and use the stump sprouts, that is juvenile material. But you're probably talking about things like doing serial grafting and things like that, where you start with a tree, you graft it, and then you cut it off and graft it again, and that's been shown to rejuvenate some woody plants. We haven't done any of that yet. But to tell you the truth, we've tried using even seedlings to start cultures from, like leaves of seedlings. And even we haven't even been able to do that. Although Elena Corredora and I'm mispronouncing her name, in Spain, she was able to start embryogenic cultures from seedling leaves of European chestnut, when we tried something similar with American chestnut seedlings, even using something as juvenile as that, we could not get it to work. So we're still stuck working with starting material that is immature seed embryos.
All right, I think we got through. Those were some excellent questions.
Yeah.
Ted Williams did have his entire body cryopreserved.
His whole body.
His whole body, but his head was removed, and it sounds like they did it separately. I'm not sure. I mean, I think it's for, you know, eventual cloning. Oh, we had another one come in, so I'm glad we talked about Ted Williams. And
Yeah.
I like Futurama. Do you ever watch Futurama? You know how they have the floating heads.
The floating heads, yes.
Yeah. Are you using this methodology to preserve germplasm of any other declining species like ash or other tree species? Ew, just ew, makes sense. No, so sorry. Are you using this methodology to preserve germplasm of other declining species?
Sorry. We don't need to talk about Ted Williams anymore. Yes, absolutely. All of the trees, we're working with lots of threatened trees, hemlocks, ash, Atlantic white cedar. We have cryostored copies of all of our embryogenic cultures from all of those trees. Some of them don't work quite as well as chestnut. We're struggling with ash a little bit. It's a little different, but yes, we have cryostored cultures of all those.
And yeah, ash is because of EAB, emerald ash borer, and there's a lot of trees that are under attack. So, you know, all these technologies have wide-ranging application for, unfortunately, the health of forests in the United States, particularly the eastern U.S., is pretty troubling. I guess I joke sometimes, maybe it's morbid, but you know, job security.
Yeah, if you're a forest pathologist or a forest entomologist, yeah, you've got lots of work to do.
Yeah, that's unfortunate.
And it's, there's a huge list of trees that are threatened because of this. And I pretty much the whole focus of my lab has shifted to threatened forest trees working. And I'm looking for collaborators, for breeders, selectors, just like, you know, the Chestnut Foundation's collaborated with me. I'm working with an ash breeder who works for the Forest Service in Ohio to send me stuff from these lingering ash trees that may be resistant. I've worked with people with hemlocks. There are some bulletproof hemlocks out there that seem to be tolerant of hemlock woolly adelgid. And so I'm looking for people to send me stuff to start cultures with, and then I will send them back the stuff we regenerate to test. So that's what we do in the lab now.
That's great. Well, maybe you can focus on Ted Williams. Anita has another great comment that Ted Williams's youngest son did it for regeneration. So I'm glad that she knows the reason why.
Okay.
Scott, thank you. This was fantastic. I really appreciate you taking the time to share with us your background. Oh, wait, I have another question. I'm sorry. There are surface chemicals that the pathogen uses to identify and infect cells. There is a metabolic system that creates these surface chemicals. It can be interrupted through genetic manipulation or chemicals that inhibit the binding. Not really a question. But
okay. So is that something we might want to look into, I guess?
I think so.
Okay. Well, thank you.
Yeah.
Pat,
Also,
did you see that from Tiago?
No.
I just want to thank my teacher, Gloria P., University of Aravo, Portugal, and informing me and my colleagues about this chestnut chat. I'm more excited about plant biotechnology. I really like this presentation. Lots of great reviews in the chat on your presentation, and I echo all that. Thanks for a very informative presentation from Anita. Thank you for the amazing presentation. So you've got some new fans out there, Scott, along with all of us at the Chestnut Foundation.
Oh, thank you.
Thank you so much.
Yeah, sorry to keep prolonging this, but Carl asked, why don't you look into the chemistry? And Meg says, implying a competitive exclusion strategy. So that's building on that other metabolomic.
I think I need some really smart collaborators to do that.
Okay, I'm out. All right, so we are taking a break in 2 weeks. That'll be Thanksgiving, so we aren't going to have a chestnut chat right after Thanksgiving. We're going to be recovering from eating too much turkey, I presume. December 11th will be our last chestnut chat for the year. We are planning on having it in 2021. We're going to move to a monthly schedule in 2021. So as soon as we know which Fridays we're going to do it on, we'll let you all know. December 11th, we're going to do a town hall. We want you to bring any and all questions. It doesn't matter what topic. Hopefully it's related to Chestnut, but maybe we can talk about Ted Williams more. But any topic, we'll have probably 5 to 10 different panelists, and we'll take your questions, whatever they are. And it'll be a really open type thing to round out Chestnut, our first season of Chestnut Chat here in 2020. Scott, thank you. I'll leave it to Lisa to have any final words.
No, just again, thank you, Scott. I bet you are a wonderful professor. This always makes me want to go back to college, even though I'm way too old to go to college.
Oh,
But anyway, it's really fun to learn these new things in these times in our lives. And everybody out there, please stay safe, wear your mask. I want to thank Steve Berlovitz, who was on the call, but he recruited Scott. And he's been recruiting a lot of really wonderful scientists to share their work with us. And I just want to commend Scott for his tenacity. Oh my gosh, how you took 10 years to get one method going. And that is just amazing to me. I guess I'm just not very patient.
oh, well, I had other things I could kind of fall back on that were working while we were, hitting the wall with Chestnut. And we didn't give up. And it's really the people in my lab that did all this. I've had those people I showed you, plus a lot of other people, just were very amazing. They worked hard and they didn't give up.
Well, that's great if you had a good team. I just want to remind everyone, I know this is shameless, but we are in the throes of our fall fundraising campaign. So please, if you have any discretionary income and you want to put us on your list of charities this year, we'd appreciate any and all amounts, and just participation is really important to us. So far, we've done really well compared to many nonprofits that are struggling, especially in the performing arts and higher ed. They're having a tough time, but luckily we have a very loyal donor base. So to those of you that do still give, thank you so much. We rely 94% on private philanthropy. So please consider that in your end of your giving. And with that, have a wonderful Thanksgiving. And we're getting through this 2020. We're getting there.
Gotcha.
Hopefully the vaccine news this week was encouraging to everybody. So take care. Thank you so much again, Scott.
Okay.
And thank you, Sarah. You do a wonderful job at facilitating and managing the questions.
Great.
Thanks,
Thank you.
everybody.
Okay, bye.
Take care. Bye.